Okay, well, thanks so much for giving me the opportunity to talk about a really fun and exciting project that we conducted the last couple of years with a really large group of collaborators, which I'll highlight a little more moving forward. And I also wanted to acknowledge from the get-go that a lot of the pictures and figures, pictures have been generously provided by Arden Blumenthal and others from the New York, New Jersey Trail Conference Dog Program, as well as others from our team. So just wanted to make sure that everybody got recognized for all their work and contributions to the project. Just really quick, for those of you on the call who don't know what the New York Invasive Species Research Institute is, it was established in 2008 to work at the interface of research and management with a mission to coordinate invasive species research to help prevent and manage the impact of invasive species in New York State and beyond. So you can see from that mission the driver behind the research project that I'm presenting today. We work closely with all the PRISM network, the DC Invasive Species Coordination Unit, and the Council, the ISAC, and IMAP, and all the others that are working hard in New York State to deal with the invasive species. I know this is probably not an audience that needs an overview of spotted lanternfly, but in case there's a sprinkling of people who aren't familiar with spotted lanternfly, it came over from Asia in 2014 into Pennsylvania. It feeds on and damages agricultural species, particularly grapes. Its honeydew excretions and the sooty mold that grows afterwards has negative impacts on fruits and leaves. Even its presence, swarming in large numbers, can keep people from wanting to go outside, canceling soccer practices, and not wanting to sit on their back deck. And there's currently research being conducted a lot of research is being conducted on spotted lanternfly biology and control, but also I'm trying to understand what its impacts are on native trees as well. So as I mentioned, it was first detected in 2014. And I just want to outline this progression a little bit because when it was introduced into Pennsylvania, the idea was that it was going to be contained and eradicated, or the hope was that it could be eradicated. But unfortunately, over the years, very few years, it really spread quickly into surrounding counties and surrounding states. But when it first came in, we were really working at the eradication containment and trying to keep it from moving farther and trying to snuff out any populations that started establishing outside the initial introduction. All the while that we're trying to contain it, there was also short and long-term solutions being utilized and under development, like new pesticides and strategic trap trees, physical barriers such as netting, sticky bands, and other physical removal, and longer-term solutions such as biological control using pathogens and parasitic wasps to reduce and control spotted lanternfly. Currently, there aren't long-term solutions available, but we're really optimistic that those will come in the future with the hard work of folks at Cornell and in other research universities. But considering there wasn't a long-term control or even a short-term control method, the strategy to reduce the impacts of spotted lanternfly in New York State was to try to keep it out or detect it early and respond and get rid of any small populations that became established. But as you all know, with early detection, it's really hard to detect things when the populations are small, and also particularly challenging with spotted lanternfly, although its adult phase is pretty flashy, its egg masses, and those are often the things that are transported first, are really pretty difficult to detect. Just to give you an example of there's spotted lanternfly egg nests on this tree, visual searches would have a hard time detecting that. They're great camouflaging here. those are the spotted lanternfly egg masses. So detection, especially when they're small populations, is really difficult. So the search was on to try to figure out the best way to detect spotted lanternfly early. We've been working, I know that New York State's been collaborating with folks down at Rutgers and other universities to look at using eDNA in a terrestrial habitat. You know, IMAP has put volunteers out on the landscape as well as PRISMs to try to be sure to report it when individuals or small populations were detected. And also detector dogs were starting to be used to see if they could help detect it either on vehicles and items coming in or in natural settings. And right around the time that all this was happening, I had the opportunity to meet Amy Hurt from Working Dogs for Conservation. It's an international program that uses detector dogs for conservation purposes, such as to try to put a stop to wildlife crime, working with invasive species, and also monitoring of rare and endangered species that are difficult to detect. And this, just a quick, brief, one moment history of conservation detection dog evolution. You know, it really is not new, but in recent years has really picked up and become more widespread, and is really starting to show its effectiveness as a tool for detecting species that are hard to find. Just some a few numbers here. You know, 5 times faster at finding brown tree snakes, 9 times more likely than camera tracks to detect single bear bobcat, 10 times faster at finding the first black-footed ferret, 16 times more areas searched for black-footed ferrets for the unit time, 36 times more likely than hair snare traps to detect a bear bobcat, and 39 times more turtles discovered per unit time. So, you know, we're getting a lot of evidence on their effectiveness. Here's just another little example from the trail conference. There's invasive species in both of these pictures, but to the human eye, that would be really difficult to find. And the dogs were able to find them. And so, you know, just even when it's not in a structured study, we can see how effective they can be. You know, another great thing that really struck me with and interested me in detection dogs, and I've seen the results of people's love for dogs in this project, that it's actually a really great outreach tool as well, because when you bring an adorable dog that's doing a great job into the picture, the media is really interested, people stop to listen, and it's a great way to engage people on invasive species and other conservation issues. So I just wanted to highlight quickly a little bit of background on the New York, New Jersey Trail Conference Conservation Dog Program. I was really excited after I met Amy Hurt from the Working Dogs for Conservation to find out that the Lower Hudson PRISM was actually developing their own conservation dogs program. It was established in 2018. And they've been using it to supplement invasive species early detection and removal activities. They can be more thorough in their removal activities. If there's like a small plant left on site, the dogs can find it. And also you can get it to be eradicated sooner. And also finding plants or species that are outside the known population or ones that were missed during removals. Some of the species they're working with are Scotch broom, slender false brome, sticky sage, spotted lanternfly, oakwill, and kudzu. So with that, the need to find early detection methods and understand what was working best for spotted lanternfly, our awesome new New York State detection dog project, and some funding from the Cornell Atkinson Center for Sustainability, which really is looking for innovative ways to address sustainability issues. And they really promote cross-disciplinary interaction and research that can be applied. So it was really a perfect fit for our project. We were thrilled to get the funding to be able to start testing some of this on the ground. We have a pretty big team here. We have folks from the New York Fish and Wildlife Co-op Research Unit, Dr. Angela Fuller and her postdoc, Ben Augustine, Dr. Ann Hayek from here at Cornell in entomology, and her postdoc at the time, Eric Clifton, our team at NISRI, the dog team, Arden Blumenthal, Josh Bies, and Linda Rollator at the time was the one that was working with us on this. Amy Hurt from Working Dogs for Conservation, Jess Cancellieri, Tom Algeyer, and Karen Snovercliff were also collaborating as we were getting this project off the ground. So we really had a real diverse and large team to address this issue. And of course, not forgetting, the main players, Dia and Fagan, who were instrumental in the research. So the study objectives were to evaluate the utility of using detection dogs as an early detection method for SLF. We were looking to compare the efficacy of human observers and detection dogs as methods to detect SLF egg masses. We narrowed it to egg masses just since this was a pilot to make sure that we were able to really get some solid results. Identify environmental factors that influence a dog or human ability to detect SLF so we can decide when and how to use the two different search methods. We're going to model the probability of an occurrence of SLF as a function of the site level landscape characteristics. And this was important because we didn't know how many SLF were out there, so we had to actually model the probability that SLF was there, and I'll explain that further, in order to then see who was more effective at detecting them. And so then it was a pilot for optimal search strategies based on our findings. And this was kind of a cool project because, and the reason why Atkinson took it on, because no other effort at the time for spotted lanternfly or any insect pest had used this modeling to understand both the probability of detecting infestations in agriculturally important areas such as vineyards and estimating detection probability of dogs and humans. So it was a unique combination of the two techniques. So just really quick, I learned a lot from this study as well, since I'm not a modeler. But what is occupancy modeling? It's a model that's used to account for imperfect detection of organisms in surveys and to determine the probability of the true presence or absence of a species at a site. This is done by quantifying the detection probability of a species at a site based off your data. And so, in your survey area, you could see a species or you cannot see the species, but if you don't see the species, you don't necessarily know that it's not there. It could just be that you didn't see it. And so that this accounts for that imperfection. This is a quick little illustration, just as you're looking at these different quadras, you go out and there's places where you're not detecting where it's present. And that this predicts the likelihood that that's occurring. So we were lucky enough to be able to find a good number of vineyards. I don't know if that's lucky, with surrounding natural areas that we could, that we knew there was some level of spotted lanternfly present. In each of the vineyards, we did 12 transects, and then we did 12 transects in the adjacent natural area, forested area, we call it. And then within the vineyard, the subunits were vines or poles, and within the forest, they were 1 meter segments of forest transect. And our study sites were pretty close to the original core because at the time, spotted lanternfly was just starting to show up very scattered in New York. So we actually had to work in New Jersey and Pennsylvania to do this work. This is just a little illustration of how the transects were set up. And our hypotheses were that occupancy of spotted lanternfly would be higher closer to forests, that the infestation level matters, that the occupancy at a transect and subunit level would be higher with a higher lanternfly infestation at a site, and the detection probability of humans and dogs should be higher at sites with higher infestation levels, which is obvious, but was important for the model. And then we statistically assigned sites to low and high infestation levels. And then finally, we hypothesized that humans would be equal to dog in vineyards, and dogs would be better in the forest since humans use visual search and dogs use their noses. So I'm just going to run through the modeling methods. We estimated lanternfly occupancy rates with imperfect detection, like I was saying, subunits for vines, pools, forest segments, 1 meter forest segments that were nested within transects. And we repeated each survey 2 times. And then we estimated transect level occupancy. So whether it was would likely to be in a transect or not, based on the habitat type, whether it was forest or vineyard, the infestation level, the interactions between habitat type and infestation level, and habitat covariates such as the topographic position index, distance to the forest for the vineyards. And then we estimated subunit level occupancy probability as a function of the same as transect level plus the subunit substrate, which would be the vine, the meadow pole, the wood pole, or other poles. And then we also modeled detection. So we estimated detection probability of the dog versus the humans, so the observer type. And then we also looked at whether there was a difference between the humans at their ability to detect and between the 2 dogs. And we looked at what it was more likely to be on the vine versus a pole versus a forest, a vineyard versus a forest, low versus high infestation levels, and the weather covariates, whether snow precipitation or wind speed would impact it. We recorded search times for each of the transects because we wanted to look at how efficient the 2 different searchers or the dogs versus the humans were. Okay. So here are the results. Vines had more spotted lanternfly egg masses than 1 meter segments of forest. Metal poles were a big attraction for spotted lanternfly egg masses. So they had higher occupancy than vines. And of course, the occupancy probability was higher at high infestation sites. And infestation effects were stronger in the vines than in the forest, so they were more impactful detection results. And so the infestations were highly infested sites obviously had higher detection probability. In the vines, the humans actually did better than dogs, but I'll talk a little bit about that later. And in the forest, the dogs did better than humans, according to the model. But we need to note here that the dogs are actually just as good at detecting in both places, but the humans are pretty terrible at finding the egg masses in thick forests. And so, which is why the humans got better rankings in the vineyards. And then the weather effects on detection by dogs. The snow, we were doing this in the winter, obviously, when the egg masses were out, but the adults were not. So the snow reduced detection probability by the dogs, and snow depth tended to be greater in the vineyard. There was a lot of snow this year. And then greater wind speeds increased detection probability by dogs, and the wind was more variable in the vineyard. So search time and search efficiency. The dogs took about 1.87 hours to search the transects in the vineyards, and the humans took 2.44 hours to search the 12 transects. In the forest, the dogs took longer, 2.08 hours, and the humans took 1.3 hours. Dogs took more time at high infestation sites, but humans did not. But that is because there's a lot of subtle details in that, as the humans didn't have to necessarily detect every single egg mass. Low infestation in vineyards, humans were slower than dogs. Low and high infestation in forests, humans were faster than dogs. In vineyards, in summary, we're more efficient than dogs, especially at the high infestation rates, and the dogs had more detections than humans in the forest. but offset that by greater search time. So they had similar efficiency. And then there were, like, as I mentioned, there were some differences between each individual observer within the dog group and within the human group, but those weren't really significant. And I just wanted to point out, in addition to like the modeling and the statistical analysis that went into comparing dogs and humans, there were a lot of observations that our entomologists made in the field. So I just wanted to kind of point out some of the observations that Eric Clifton had made while he was out there doing the surveys. And if you're searching for spotted lanternfly, the undersides of cordons, tight spaces between the trunks and the support, and the trunk bases were really preferred locations for spotted lanternfly to lay their eggs. Undersides of cordons, as you can see here, we've got a lot of great photos from the field. Here's the tight spaces between trunks. And at the base, they were really common, and especially even under the grass here. So if you're looking at low infestation sites, those are great places to search. And so also we really found that the best place to search for spotted lanternflies are the areas closest to the forest and also the trees that are at higher elevations next to the vineyard. This is just a tree with many spotted lanternfly egg masses that was right next to the edge of a vineyard, and most of them are on the underside. And this is our findings in that were supported by work that was done down in Pennsylvania by Heather Leach, where 44% of spotted lanternfly egg masses were found within 15 meters of the vineyard edge. So just another note from Eric here that the undersides of leaning or fallen trees around the property are a really hot spot. And he also just a quick observation that he found that mantids were he was thinking that mantids were actually attacking spotted lanternfly in the vineyards. And it's just an interesting observation to keep an eye out for. But I don't know that that would provide any sort of population level control, but just an interesting interaction. So in summary, it's easier to obviously to excuse me, to detect spotted lanternfly at high infestation sites. Infestation level was influenced by detection probability, more in vineyards than in forests. Greater difference in detection between high and low infestation sites. Vines had higher occupancy than forests. Meadow poles had higher occupancy than vines. And humans detect spotted lanternfly in vineyards, more spotted lanternfly in vineyards than dogs, but have greater search time. And dogs detect more spotted lanternfly in forests than humans, but similar efficiency when accounting for search time. I don't know why that happened, but some of the challenges, this was the logistical challenges of working in COVID. It was really in 2020 that a lot of this work was taking place, really got us having to be creative. Weather and the difficulty to search and record data in deep snow was a challenge that we had to end up having extra folks on, and then extra cars. We were trying to align the search strategies with the way dogs would search in a real search scenario, was also a challenge because we couldn't exactly match that within the transect methodology that we were using. And just little surprises like for us non-vineyard owners that winter pruning takes place, and trying to manage with that. And I have to give a shout out to Arden who dealt with all of these coordination dilemmas very well and really helped us get through. But really the strength of a resilient and collaborative group like this and with diverse expertise really helped to overcome a lot of these challenges. So just wrapping up, dogs can be more effective than human searchers at detecting spotted lanternfly egg masses, but it's really context specific and requires additional research. This study can be served as a pilot for how detection dogs and occupancy modeling can be applied to address the complex task of early detection of invasive species. And then the results from this can inform search strategies that New York State employs for early detection by understanding influences of weather and context on detection probability. And just really the value of modeling occupancy for spotted lanternfly that accounts for the probability of detection using naturally occurring egg masses can establish a network that we can use for dogs for searching for other taxa as well, whether they're invasive, threatened, or endangered in diverse habitats and geographic locations. And multi-scale occupancy models allow occupancy to be estimated at the transect level and the subunit level, and the detection to vary as a function of observer, infestation level, and weather covariates. And we could really use this framework for any invasive species. So moving forward, just a few tips. Search vineyards close to forests. Employ detection dogs in forest sites. They're 2 times greater detection based on our study. Humans can search vineyards pretty effectively, so we could do that as well if we didn't have dogs. Just for vineyard owners, metal poles were a huge attractor for spotted lanternfly to lay eggs. So maybe wooden poles would be less attractive in drawing them in. And but actually, we need to consider what a real early detection rapid response scenario would look like, where every egg mass doesn't have to be accounted for. So, within our results, that the reality of how we would search couldn't exactly be replicated. So just a big thanks again to everybody that was involved in this project, to the Atkinson for the funding. Thanks for all the slides and photos that came from these folks. And also to all the vineyards who let us romp around their properties multiple times with dogs and many humans, and for really trying to adapt their pruning strategies to our sampling. And so right now we're just finalizing the data analysis and really refining recommendations to make sure they're meaningful for on-the-ground needs and search strategies. We'll be publishing this hopefully in the next couple of months. And we'll be providing a more of a formal recommendation to folks that would be interested. So that's it.
All right, thank you, Carrie. A few things